This conference will now be recorded.
Sorry, that got disconnected. So we were saying about the genetic code. So we had learned about 4 bases and 20 amino acids. Then the genetic code. We had learned about the features of that genetic code. Then that codon is in triplet form. Different features of genetic code we have learned. So codon means that is in triplet form, and with different combinations. In different combinations, these 4 bases will join, and that forms 64 different codons. So how many codons? We have totally 64 codons. So among these 64 codons, 61 codons that codes for amino acid, and 3 codons do not code for any amino acid. So these codons which are not coding for amino acid, they are called as stop codons. So the codons which are not coding for any amino acid. So when there is a amino acid, there is a codon, stop codon, which is not coding for amino acid, this amino acid synthesis or this synthesis is getting stopped. So that acts as a stop codon. That's how we are calling as stop codon. So the code is universal. You know that these codes are universal. And some amino acids, we have already discussed all these points. Some amino acids are coded by more than one codon, right? The table we have discussed in the last class about different amino acids and different combinations of each base and how it is coding for amino acid. Everything we have learned. So general features, I'm saying about the features of genetic code. So there are totally 64 codons, and among that 61 codons they code for amino acid, and another 3 codons they do not code for any kind of amino acid. So those codons we are calling those codons as stop codons, and these codes are universal, and some amino acids are coding by more than one codon. Okay, so different combinations. Among that, some different combinations, they code for the same amino acid. Okay, so without any full stop or any separation, so without any kind of separation, this codon, it is read in mRNA. So mRNA is reading these codons without any separation, or there is no separation between each codon. So it is read continuously. That means the messenger RNA is reading these codes continuously. Okay, so the AUG is coding for methionine, and this is also acting as AUG codes for methionine, and this methionine that will act as a start codon or an initiator codon. Okay, so methionine, it works normally, and that is having an extra role that is called as a start codon or an initiator codon. Okay, like stop codon, starting codons are also there. So methionine is acting as the starting codon or an initiator codon also. Okay. So the genetic code. Then which are those 3 stop codons? Methionine, AUG, that is the starting code. Okay, starting genetic code, initiator genetic code. Then the stop codon. We should learn about the stop codons also, that do not code for any kind of amino acid. These stop codons will never code for any kind of amino acid. Which are they? UAA, UAG, and UGA. Which are the stop codons? UAA, UAG, and UGA. So these combination, whenever these combination of bases comes, that don't code for any kind of amino acid, and these three codons, the triplet codons, they are called as stop codons because they won't code for any kind of amino acid. Then we learned about mutation of this genetic codon, right? So if mutation happens, there will be some kind of changes in the amino acid sequence also. And therefore, the final protein, that final protein expression, that also may change, or that creates some kind of changes in the final protein expression also. Okay. The formation of that protein, that there will be some kind of changes from the normal protein. There will be some kind of changes in the formed protein when mutation is there. That may be addition or deletion. When one genetic or one base is removed from that, that forms one type of protein. From the normal protein, that shows a kind of difference. When something is added, some base is added to that, then also that protein differs from the normal one. So either it may be addition or deletion, whatever may be, that creates some kind of changes in the final protein expression, or the amino acid sequence itself varies, and that forms some kind of protein expression in different way. From the normal state, it is showing some kind of difference. Then, in today's class, we are going to discuss about tRNA. Okay. So what is tRNA? So while discussing about different types of RNA, I had just mentioned about the tRNA also. Hope you remember that. So what is tRNA? tRNA or transfer RNA. It is the type of RNA that helps in the synthesis of protein. From where? From mRNA. That helps the protein synthesis of protein from mRNA. So what is tRNA? tRNA or transfer RNA that helps in the synthesis of protein from mRNA. So how it helps? How this tRNA helps? So this tRNA that will function as adapter molecule. tRNA, you know that tRNA functions as adapter molecule during protein synthesis or during this translation process. So during that time, this tRNA works, or that functions as adapter molecule. Okay. So during earlier period, they called this tRNA as sRNA or soluble RNA. Okay, because they were not knowing the exact function of that. So nowadays it's very clear that tRNA, it is working as adapter. It is working as an adapter. And how it is working as an adapter, or how it works? It links the amino acid to the nucleic acid. The tRNA links the amino acid to the nucleic acid. Here, the amino acid is linking. It is getting linked to the nucleic acid with the help of this tRNA or the transfer RNA. So that links the amino acid to the nucleic acid. Then tRNA carries the amino acid. This tRNA carries the amino acid, and this amino acid which is to be added in the peptide chain. Okay. The whole procedure I'll explain later, and very clearly, step by step I'll explain. So the tRNA, what is the role of tRNA? tRNA carries the amino acid. The tRNA that carries the amino acid, and this amino acid, which amino acid it is carrying, which is to be added in the peptide chain. Okay. And that tRNA also decodes or deciphers the codon for that particular amino acid in the mRNA molecule. So that's how it works. So how it is working as an adapter? It links the amino acid to the nucleic acid. Okay. This tRNA links the amino acid to the nucleic acid. Okay, this is the nucleic acid. Here you can see that nucleic acid and the amino acid is getting linked with the help of this tRNA. So the tRNA carries the amino acid which is to be added in the peptide chain, and thus the tRNA also decodes or deciphers the codon for the amino acid in the mRNA molecule. Okay, and as the name indicates, from this tRNA or transfer RNA, from the name itself, it's very clear that transferring the RNA, or it is a transfer RNA that works for transferring the amino acid. So what is the role? From the name itself, it's clear, or as the name indicates, tRNA is, or transfer RNA, it is transferring the amino acids. Okay, so tRNA is transferring the amino acid. So now we can learn about the structure of that tRNA. Without learning the structure, we can't make out how it is working for protein synthesis. So let's learn about the structure of tRNA. Here in this picture, you can see here, this picture is there. Here you can see the red colored line. This is the mRNA or the messenger RNA. Messenger RNA from 5' to 3' direction, right? You can see that red color line, which is the mRNA from 5' to 3'. Okay, so in that you can see some codon. Right, here you can see some codon. So for an example, just to explain how it works in a particular part, and how it is coding. So for that, here you can see the codon. So we are selecting a particular part here. From this mRNA, we are selecting one region, only one part we are selecting, and one codon we are selecting from this mRNA. Okay, like this, so many are there on this mRNA, but we are selecting, just for explaining, we are selecting only this part. Don't get confused that there will be only one codon in one mRNA. Don't get confused like that. Here, one part, particular part I am explaining. Okay, so the codon here, here this marked one is AGU. So you can see that the tRNA, which is having anticodon loop. Here, this is the tRNA, and this loop, this is called as the anticodon loop. This is the tRNA. Here, first we explained about the mRNA, this red color line, that is the mRNA, and from mRNA we selected one particular portion with codon, that is AGU. Now we have selected the codon AGU. Next, the top part, this is the tRNA, and the tRNA is having anticodon loop. Okay, for this codon there is particular opposite codon or anticodon. So in this loop, that anticodon will be there. In this loop kind of portion, there will be the anticodon. And in this anticodon loop, you can see that it's having bases, which is complementary, and these bases are complementary to this codon, right? A pairs with U. Why? In DNA, what we have learned? A and T. Adenine will pair with thymine, right? And guanine with cytosine. But in the case of RNA, adenine pairs with uracil. That don't get confused. In RNA, that pairing is with uracil. Here also you can see A and U. Here A and U. So adenine is not with thymine, it's with uracil. In the case of RNA, adenine and uracil will pair. Okay. So this anticodon loop you can see here, and you can see that it is having bases, and these bases are complementary to this codon, this complementary bases. Here you can see the complementary base pairing or the anticodon. Okay. So that is the anticodon loop of tRNA, and the basis of the anticodon loop of tRNA is complementary to the codon, right? So this tRNA is also having amino acid acceptor end. Okay, here this tRNA, it is having amino acid acceptor end. So what does it do? This amino acid acceptor end that will bind to the amino acid, and you know each amino acid is having a specific tRNA. Here you can see the amino acid, it is bound to this tRNA. So this amino acid, each amino acid is having specific tRNA. So this is serine. It is having specific tRNA. Here this is another amino acid that is having separate tRNA. So each amino acid is having specific tRNA. And there is another specific tRNA that is called as initiated tRNA. Why it is called as initiated tRNA? Note down this. This is very important for NEET exam. Initiated tRNA, that is very specific tRNA, that is initiated tRNA. Why it is called as initiated tRNA? Because that initiates the process. Okay, the process of protein synthesis. It is initiated by the initiated tRNA. And we have already told that stop codon that doesn't have any tRNA. Stop codons won't form any tRNA, right? So let's see the tRNA that it has an anti-codon loop and that has bases complementary to the code. Here, this point we have explained now. This tRNA, it is having anti-codon loop, and that anti-codon loop that has bases which is complementary to the code. Okay, and it also has an amino acid acceptor end to which it binds to amino acid. There is one amino acid acceptor end and that is binding to the amino acid. Then tRNA are specific for each amino acid. Each amino acid is having specific tRNA, and for initiation there is another specific tRNA, and this specific tRNA is called the initiated tRNA. So there is another specific tRNA which is called as initiated tRNA, and this initiated tRNA is starting, or that is helping for the initiation of the process. Okay, and there are no tRNA for stop codon because there is no need of protein synthesis. Stop codon, that is stopping. There is no continuous process, so it is stopping the process. So there is no need of any tRNA. So there are no tRNA for the stop codon. Next, here you can see the structure of tRNA. So in the actual structure, the tRNA, here you can see it is like a complex molecule, right? It's like inverted L. It's having inverted L shape. Note down all this. These are important points for NEET MCQ, NEET exam. So it's very important. It's having the actual tRNA. It's having complex structure and it is like inverted L. You can see L, inverted L. See, this is having inverted L. Inverted L shape. See, in the first picture you can see. But look at the second picture. Here, this one. In the second picture, you can see the secondary structure of tRNA. This is the secondary structure of tRNA. How does it look? The secondary structure of tRNA looks like a clover leaf, right? It's like a clover leaf structure, or that is the secondary structure. Secondary structure of tRNA. So the secondary structure of tRNA looks like a clover leaf, and this is also called as clover leaf model. Okay, secondary structure of tRNA is called as clover leaf model, and scientist named Hawley proposed the clover leaf model of tRNA, and this is the secondary structure. And in the primary structure or in the actual structure, you can see that is a very complex structure and it is in the form of inverted L. But the secondary structure, it is like a clover shape, or it is called as clover leaf model. So scientist named Hawley, he proposed this model, and it's very important, this part, this structure of tRNA, that is very important for your NEET exam. And this one, the structure of this clover leaf model, this is important for your NEET exam and for your board exam. You will get a question to draw the secondary structure of tRNA and to mark the points, right? So just learn this structure, clover leaf model. It's very important. It is a secondary structure and it is in clover shape, and scientist who proposed, Hawley. Scientist Hawley, he proposed this model. Then, now we'll discuss about the function of this tRNA. tRNA functions. Okay. So tRNA, it is having very important role in protein synthesis. The main thing is tRNA is having very important role in the synthesis of protein. So this tRNA, that is acting as adapter molecule. We have already told that is acting as an adapter molecule. For what it is working as an adapter molecule, or how it is working as an adapter molecule? That this tRNA acts as an adapter molecule for linking amino acid to the specific codon, and that codon which is present in mRNA. Okay, so how it is working? It works, or it acts as an adapter molecule for linking amino acid to the specific codon which is present in the mRNA. Okay, it is linking. See here, we learned about this from this picture. It's very clear. So how tRNA is working? It acts as an adapter molecule for linking the amino acid to the specific codon, which is present on the mRNA. Here this mRNA is present, on that specific codon is present. So this tRNA is linking specific amino acid to the codon which is present on the mRNA. Okay. So the first step of protein synthesis is amino acylation. Note down, first step of protein synthesis is amino acylation. That is the first step in the protein synthesis. So I have already told, for each amino acid there is a specific tRNA, right? For each amino acid there is a specific tRNA. So what this tRNA or transfer RNA will do? This tRNA or this transfer RNA that carries amino acid during translation time, right? The tRNA carries amino acid during translation time, and to where this tRNA transfers the amino acid unit? To the ribosomal subunits. So the tRNA carries amino acid in the time of translation process to the ribosomal subunits. Got it? To the ribosomal subunit, the tRNA carries the amino acid. So to the ribosomal subunit, and the ribosome is the site for protein synthesis. Correct? Ribosome, there only the protein synthesis, or the process of protein synthesis is mainly taking place in the ribosome. So the tRNA transfers the amino acid to the growing polypeptide chain. The tRNA that is transferring the amino acid to the growing polypeptide chain. Okay, here you can see this is the structure, and here this is the growing polypeptide chain. Here you can see. So the tRNA that is transferring the amino acid. This tRNA will transfer the amino acid to the growing polypeptide chain. So this polypeptide chain that is present on the polypeptide chain is present in the ribosome. So how it works? How it is working? The tRNA that is transferring the amino acid to the growing polypeptide chain in the ribosome. And that's how this protein synthesis is taking place. Okay. So this ribosome, that particular ribosome, it is having 3 binding sites for tRNA. The ribosome is having 3 binding sites for the tRNA. Okay. So which are those 3 sites? A, P and E. We'll mark it as A, P and E. A means amino acyl site. P is peptidyl site and E is exit. Okay. So there are 3 sites, 3 binding sites. The ribosome, it is having 3 binding sites for tRNA, A, P and E. A stands for amino acyl site and P is for peptidyl site and E is exit. Okay. So the decoding. Next, the decoding of codon of this mRNA. Okay. The decoding of codon of mRNA by a specific tRNA, that will continue. Up to which level? Until the entire sequence of a polypeptide chain is translated. Okay. There is a polypeptide chain. So until that whole polypeptide chain is getting translated, the decoding of this mRNA, that specific mRNA, with the help of that tRNA, that will continue. The whole sequence of that polypeptide chain is getting over means, or when it is getting over, that time this decoding of this codons of mRNA will get stopped. Okay. So with the help of this tRNA, it is taking place, this decoding. So whenever that complete polypeptide chain is getting translated, that time this decoding will get stopped until and unless it will work. So that will continue the work up to complete translation. Got it? So the tRNA, function of tRNA. tRNA plays it is playing an important role in the protein synthesis, and that acts as an adapter molecule for linking amino acids to its specific codon present in mRNA. Okay. The first step in protein synthesis is amino acylation. Note down, first step is amino acylation. So for each amino acid, there is a specific tRNA, and this tRNA carries amino acid during the translation process in the ribosomal subunit. Okay. Then what happens? Then the tRNA transfers the amino acid to the growing polypeptide chain in the ribosome. And this is having 3 binding sites for tRNA, namely A, amino acyl site, and P, peptidyl site, and E is the exit. Okay. So until the entire sequence of a polypeptide chain is translated, this decoding process or the decoding of these codons of mRNA continues. How it is getting decoded? With the help of specific tRNA. Got it? Next, we are moving to an important topic that is called as translation or protein synthesis. Okay, this is very important topic for NEET and board exam. So note down each and every point. The translation. What do you mean by translation? Translation means the synthesis of protein, or that is the process of protein synthesis. That means it is nothing but the polymerization of amino acid. See, so many amino acid units will come together and that forms the protein, right? Amino acid, that is the building blocks of protein. So, polymerization of amino acid to form polypeptide, that is called as translation. So, the order and sequence of this amino acid, how it is getting decided, who decides, or how it is getting decided, how we can decide the order and the sequence of that amino acid. We said there are so many amino acids and the polymerization of these amino acids, that is the protein synthesis we said. So how the order and sequence of amino acid that is getting decided? That is the question. So how it is getting decided? It is based on the sequence of base. Sequence of base in the mRNA. So based on the sequence which is present in the mRNA, the sequence of amino acids we can define. Okay. Here sequence, one particular sequence of mRNA is present. So how we can decide the sequence of amino acid? Based on the bases which are present in the mRNA. Definitely, see here the sequence, you know the sequence of this mRNA. You can easily find out the opposite sequence, right? And from that, which one, which amino acid is forming, or that codes for which amino acid, that is also easy to find out, right? So the amino acids, they are joined by a bond, and that bond is called as peptide bond. Amino acids are joined by the bond which is called as peptide bond. No doubt, amino acids are joined by peptide bond, and this peptide bond formation that needs energy. Peptide bond formation needs energy. So amino acids are joined by peptide bond, and the peptide bond, that peptide bond formation needs energy. So in the first stage itself, in the presence of ATP, you know, ATP is the source of energy, right? So in the first stage itself, in the presence of ATP, these amino acids are getting activated. Amino acids will get energy from that ATP, and these amino acids are linked to their particular tRNA. Okay, these amino acids are getting energy from ATP and they are getting activated, and these amino acids are linked to particular tRNA. Then these are called as, or this particular step that is called as amino acylation. I said that is a starting stage of protein synthesis or translation, starting step of protein synthesis, amino acylation. So how it is? In the presence of ATP, these amino acids are getting activated, and these amino acids, they are linked to the particular tRNA. Okay, so this is called as amino acylation or charging of tRNA. No doubt this step is called as amino acylation or charging of tRNA. Okay. The translation means the process of polymerization of amino acid to form a polypeptide. And the order and sequence of amino acids are defined by the sequence of bases in the mRNA. How that mRNA base sequence, exactly based on that we can decide the, or we can predict the sequence of amino acid. And the amino acids are joined by a bond. That bond is known as peptide bond. Amino acids are joined by a particular bond. That bond is called as a peptide bond. And the formation of a peptide bond, the formation of this particular peptide bond, that needs energy. That needs energy, and the peptide bond formation that requires energy. And the first phase, in the first phase, the amino acids are activated in the presence of ATP. So in the presence of ATP, the amino acids are getting activated, and they link to their tRNA, specific tRNA. Okay, amino acids will get activated, and these amino acids are linked to that specific tRNA. So the process is commonly, this process is commonly called as charging of tRNA or amino acylation of tRNA, which is the starting stage of translation or protein synthesis. Protein synthesis, that process is called as translation. Okay. Next, see here in this picture you can see two charged tRNAs. They are brought nearby. Here this tRNA and this tRNA, they are brought nearby, and the peptide bond formation between them will be easy. Okay. Here, when two charged tRNA, they are brought together, if they are very near, that peptide bond formation of that particular peptide bond will be very easy. Okay. If the distance is more, it will become difficult. So when it is brought nearby, the peptide bond formation between them will be favored energetically. Okay. So it will be easy. And the presence of a catalyst, if there is a catalyst, that enhances the rate of peptide bond formation. Everyone know, you people know the role of a catalyst, right? Or how a catalyst is functioning. In an enzymatic reaction, the catalyst, how a catalyst will work? So the presence of a catalyst that would enhance the rate, that enhances the rate of that peptide bond formation. Okay. So here whenever these two tRNA, those are brought nearby, two charged tRNAs are brought nearby, that favors the peptide bond formation, and the presence of catalyst that increases the rate of peptide bond formation. Got it? Then, the synthesis of protein. You know that protein synthesis is taking place at ribosome. So the ribosome is having structural RNA. So that ribosome consists of structural RNA, and it is having around 80 different proteins. Okay. So the ribosome consists of structural RNA and about 80 different proteins. And when a ribosome, it is in an inactive stage, that exists as two subunits. Here you can see this green, the whole thing, that is a ribosome. Okay. So here ribosome is having two subunits. So when it is in inactive stage, that forms or that exists as two subunits. Here you can see the bigger one or the larger subunit and the smaller subunit. You can see for this ribosome, the upper one, that is the larger subunit, and the down one, this is the smaller subunit. When it is in inactive stage, that exists as two subunits, a larger subunit and a smaller subunit. So when that smaller subunit that encounters an mRNA, that process of translation or the protein synthesis, that is the beginning stage of that protein synthesis. See, when that smaller, here you can see this is the smaller subunit. When this smaller subunit encounters the mRNA, here you can see in the smaller subunit, this mRNA is present. So whenever the smaller subunit encounters an mRNA, that time the process of mRNA, this mRNA translation, the translation of mRNA into protein, that procedure or that process is starting at that stage. Okay. So there are two sites in the large subunit for the binding of amino acid. Okay. So when that amino acid binding, it's very near, or that those two units are very close, then the formation of peptide bond, it is taking place, or that is favored when it is near or it is close enough to each other, means that there the formation of peptide bond, that is very easy. And this ribosome, that is having another role, that acts as a catalyst for the formation of this peptide bond. Ribosome acts as a catalyst for the formation of the peptide bond. Okay. Then in the translation, another point is this translational unit in mRNA. See, in a translation unit in mRNA, there is a start codon and a stop codon. It is flanked by a start codon and a stop codon. Okay, which is a starting codon AUG. It codes for methionine, that is acting as a start codon. So in a sequence of mRNA, there will be a start codon and a stop codon. Okay, so in that mRNA, you can see some additional sequence. Note on all these points, it's very important. So in an mRNA which is ready for or which is working as a translational unit, in that particular mRNA, the sequence of RNA, in that sequence of RNA, it is flanked by the start codon and the stop codon. Okay, and that mRNA that also has some additional sequence. That mRNA is having some additional sequence. These are not translated, and these untranslated regions, UTR, these are untranslated. Not the whole, that mRNA that is having some extra regions which is not at all getting translated. So that regions are called as untranslated regions or UTR. Normally we'll mention as UTR, the untranslated regions. In mRNA, some additional sequences will be there which are not getting translated at all. So these particular regions or that part is referred as untranslated region, UTR. UTR or untranslated region, no doubt. So the UTRs, they are present at 5' end and 3' end. In 5' end, near to 5' end, before the start codon, it will be present. So the UTR will be present before the start codon, after the stop codon. So before the start codon, UTR will be present, and after the stop codon, in the 3' end, after the stop codon, it will be present. So stop codon means there that process will get stopped. So after that UTR is present means that won't get translated, right? These are untranslated regions. So before the starting codon means in this 5' end, before the starting codon that UTR is present means from the start codon only that protein synthesis or the translation process is starting, right? That is the starting stage. So before that UTR will be present because that portion is not getting translated. Got it? But these UTRs, they are important because they are efficient. For an efficient translation process, this is required. Okay, so they are very important. UTR and untranslated regions. Untranslated regions are important. They are required for efficient translation process. Got it? Next. So for initiation, the ribosome binds to the mRNA at the start codon. During starting time, starting of the translation, the ribosome, where it will bind? At the start codon. Because from there, this ribosome will bind at the start codon. That start codon, obviously from that start codon only that translation process will get started, right? That is recognized by initiated tRNA. That initiated tRNA will recognize the start codon and the ribosome binds to the mRNA at the start codon. Okay, so the tRNA, initiated tRNA is recognizing the, among all these codons, the initiated tRNA, it is recognizing the start codon. Okay, this AUG. So the initiator tRNA is recognizing the start codon, and at that start codon, this mRNA, that ribosome binds to the mRNA at that particular start codon. So the start codon which is present on that mRNA is recognized by the initiated tRNA, and at that particular site this ribosome binds. Okay. Then after binding, this ribosome proceeds to elongation phase. So that is the starting, initiation. Next is elongation. So the ribosome proceeds to the elongation phase for protein synthesis. Ribosome, next it is moving to the elongation phase for the synthesis of protein. So during elongation phase, complex is composed of an amino acid. See, different amino acid complexes will form. Those complexes link to the tRNA. That will get linked to the tRNA and sequentially bind to the appropriate codon in the mRNA. Okay. So how it will work? That forms the complementary base pair with the tRNA anticodon. With the tRNA anticodon, that makes the complementary base pairing, and like that, see, you can see that a bead on a thread, it is moving forward, means how it will be like that. On this mRNA, this ribosome is moving. Okay. So the ribosome is moving from codon to codon. First, one codon it will finish, then it will move to another codon, then to next codon. Through that mRNA, this ribosome is moving from codon to codon. Okay. And it is reading all this. So amino acids are added one by one. After reading, it will form the amino acid and it is added one by one. After each codon, it is forming an amino acid and it is adding. Okay. So each amino acid is added one by one and it is translated to polypeptide sequence. Here you can see. When this ribosome is moving, that forms the amino acid and it is adding to the polypeptide chain, this growing polypeptide chain, and at the end of that process, that releases a factor. That factor which is binds to the stop codon. So that there comes the termination. Okay. So terminating that translation and thereby releasing the complete polypeptide from the ribosome. So there comes the termination of that translation. So at the end of this growing polypeptide chain formation or that process, a release factor binds to the stop codon. At the stop codon, a release factor is getting bound to that stop codon and terminating the translation. That release factor that is getting bound to that stop codon and that thereby it is getting terminated. Okay, that stop codon, that means the signal to stop this translation or the protein synthesis. So it is getting stopped there. Then that is the termination or the end of that process. Then it is getting released. The complete polypeptide, that complete growing polypeptide chain, that process goes over stop codon, that found the stop codon, and that is the end of that process after reaching the stop codon. So next is the release of this polypeptide from the ribosome. This ribosome releases the polypeptide chain. Got it? So that is the whole process of protein synthesis or the translation process. So for initiation, the ribosome binds to the mRNA at the start codon and that is recognized only by the initiator tRNA. Then this ribosome proceeds to the elongation phase of protein synthesis, and in the elongation phase, the complex is composed of an amino acid linked to the tRNA. Then sequentially bind to the appropriate codon in mRNA by forming complementary base pair with the tRNA anticodon. Okay. Then that ribosome moves from codon to codon along the mRNA. Then these amino acids, they are added one by one and it is translated to polypeptide sequence. Then at the end, a release factor binds to the stop codon. To the stop codon, a release factor is coming and it is getting attached. So the terminating translation and releasing the complete, that terminates the translation process and that releases the complete polypeptide from the ribosome. Got it? So the polypeptide chain is getting released and now it's ready for the expression. Got it? Is it clear? Do you have any doubt in this protein synthesis translation process? You want me to repeat? Deepa, is it clear? So hope up to this everything is clear. Just go through this. If you have any doubt, you can ask me in the next class. Okay, thank you.